Good afternoon everyone, how are you all? So today we will start respiration in plants. Yes, so we are going to solve the PYQs. Along with that we are going to solve the concept along with the PYQs. So our first PYQ is, given below are two statements, one is labelled as assertion and another one is labelled as reason. Try solving this question. Assertion: ATP is used at two steps in glycolysis. Is it correct? Yes, ATP is used in two steps in glycolysis. Then first ATP is used in converting glucose into glucose 6-phosphate, correct, and the second ATP is used in conversion of fructose 6-phosphate to fructose 1,6-bisphosphate or diphosphate. Is it correct? Yes, this is also correct. So if we say ATP is used at two steps in glycolysis because first step is used in the conversion of glucose into glucose 6-phosphate and second ATP is used in conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. Yes, this is actually the reason where and all it is using. So we can say B for Bombay is a correct answer. Both A and R are true and R is the correct explanation of A. Is it correct? Yes. So we will move on to the second question of today's. So what is the net gain of ATP when each molecule of glucose is converted to 2 molecules of pyruvic acid? Each molecule of glucose enter into glycolysis, the end product is 2 molecules of pyruvate. Glucose is 6 carbon molecule, the pyruvate is 3 carbon molecule. So 2 molecules of pyruvate, 3 carbon compound will be formed. In that we need to find the net gain of ATP, only ATP, that is substrate level phosphorylation we need to say. Substrate level phosphorylation. That is in the last step when phosphoenol pyruvate will be converted into pyruvic acid, that is pyruvate, 2 molecules of ADP will be converted to 2 molecules of ATP. So what is the net gain of ATP? It is this 2, right? Any doubts, children? Any doubts? Answer to the question. Are we solving together? Are we solving together? Yes, Sangeeta. In the whole process, 2 molecules of NADH plus H plus will be produced and 4 molecule of ATP will be produced and 2 molecule of ATP will be utilized. Out of all this, if we minus this, we will end up in 2 molecule of ATP at the last. So the net ATP, we are finding the net ATP. So the net ATP is only 2 molecules of ATP, that too substrate level phosphorylation. This 2 molecule of NADH plus H plus, as this is a reducing equivalent will enter ETC, isn't it? So we are not considering this, only the net ATP will be 2 molecule of ATP, that is in the direct ATP which is synthesized in glycolysis, that is 2 molecule of ATP in substrate level phosphorylation, that is direct ATP which can be utilized there, then and there, wherever it is needed, that it will be utilized there. Understood? What amount of energy is released from glucose during lactic acid fermentation? What amount of energy is released from glucose during lactic acid fermentation? It is actually less than 7%. Less than 7%, because we don't say it is total oxidation, right? It is a partial oxidation. It is partial oxidation, where pyruvate, in the lack of oxygen, especially in the muscle cell, it will be converted to lactate. Here, in this process, actually, whatever the molecule of this one, NADH2 will be, which will be produced, it will be utilized. NAD, it will become NAD plus, isn't it? So, the lactate is actually 3 carbon compound. Here, the glucose, which was 6 carbon compound, it got partially oxidized to 3 carbon compound pyruvate, again to the lactate or lactic acid, that is 3 carbon compound. Is it total oxidation has taken place? Like the glucose has 6 carbons, right? Only breakage of this bond has taken place. There is no breakage of all the bonds, which means it is a partial oxidation. Partial oxidation. Total amount of energy they will be not getting. It will be less than 7% of energy it will be released in lactic acid fermentation. Understood? Conversion of glucose to glucose 6-phosphate. The first irreversible reaction of the glycolysis is catalyzed by glucose to glucose 6-phosphate. It is catalyzed by hexokinase. Hexokinase is the enzyme. Kinase. What kinase do? Actually transfers the phosphate molecule. Okay? Please try answering. So, it is glucose, which is a hexose. The first step is, it is irreversible step. It will be converted to glucose 6-phosphate in the presence of hexokinase enzyme, where one molecule of ATP will become ADP, adenosine triphosphate, 3-phosphate group. It will adenosine diphosphate will be 2-phosphate group. 1 phosphate group will be transferred to glucose, which becomes glucose 6-phosphate. The first reaction in the glycolysis, and this step is irreversible step. This step of the glycolysis is irreversible step, and it is catalyzed by hexokinase. In which of the following process CO2 is not released? In aerobic respiration in plants, yes, CO2 is released. The glucose, actually, when it is getting oxidized, all the carbon molecules will be evolved through CO2 only. Aerobic respiration in animals. Aerobic respiration में भी CO2 release होगा ना. CO2 will be released. Alcoholic fermentation में the last step, it will become the acetaldehyde will be converted to ethanol. Before that, pyruvate which is converted to acetaldehyde, here decarboxylation takes place, that is CO2 will be evolved. This also evolves CO2, alcoholic fermentation. Only in the lactic acid fermentation. In the lactic acid fermentation, what happens? Pyruvate pyruvate, that is 3 carbon molecule, will be converted to lactate, isn't it? Here this is also 3 carbon, this is also 3 carbon compound. So there is no release of CO2, in which the lactic acid fermentation, the lactate fermentation is in which the CO2 is not released, isn't it? So aerobic respiration in plants, aerobic respiration in animals, here both glycolysis is taking place in cytosol and Krebs cycle taking place in mitochondria, almost same. There is no difference, it is almost same, everything will be same. So both the ways the respiration, carbon dioxide will be released aerobic mein, and alcohol fermentation mein it is also carbon dioxide is released, and only in the lactic acid fermentation carbon dioxide is not released, because pyruvate is also 3 carbon, lactate is also 3 carbon. Where the carbon dioxide is released or any carbon is released from here? Nowhere, right? Lactic acid fermentation. Aerobic respiration, match the list 1 and list 2. Choose the correct answer from the options given below. Okay, we need to find about the complex of electron transport system. Complex 1 we call it as, it is, the complex 1 is NADH dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3 is, the complex 3 is cytochrome reductase, which is BC1, and complex 4 is cytochrome C oxidase. Yes, so it is A1 B4 C2 D3. So B for Bombay, this one is the correct answer. Any doubts till now? Any doubts till now? Which of the following are correct about cellular respiration? Cellular respiration is the breaking of carbon bonds of the complex organic molecule by oxidation. Yes, this is correct. The entire cellular respiration takes place in mitochondria. Is it correct? Entire thing is taking place in mitochondria? No. Glycolysis is taking place in cytoplasm, and Krebs cycle, link reaction and Krebs cycle are taking place in mitochondria. So which means B is wrong. Where B is wrong, so we can eliminate it by telling these are wrong. We will go through the other statement as well. The fermentation takes place under the anaerobic condition in germinating seeds. Yes, it takes place. The fate of pyruvate formed during the glycolysis depends on type of organism. Fate of pyruvate, yes, if they are anaerobes, pyruvate will be converted to ethanol plus carbon dioxide, isn't it? So yes, if they are aerobes, the pyruvate, if there is aerobes, the pyruvate will be converted to acetyl CoA in link reaction, and it will enter into the Krebs cycle. So yes, it depends on the types of organism, whether they are aerobes or anaerobes. So this is also correct. Water is formed during respiration as a result of O2 accepting electrons and getting reduced. At the end, the terminal electron acceptor, O2 we call it as terminal electron acceptor. What happens at the end? 2 electrons will be coming from the ETC, that will be accepted by oxygen with the help of Also with the help of the protons which are there in the matrix, it will be converted to H2O. This will be the final step. After complex 4, that is after complex 4 this happens, complex 4. Yes, so water is formed during the respiration as a result of O2 accepting the electron and getting reduced. This is also true. That means A, C, D, E are true and B is false. With the elimination technique we can definitely eliminate B, C and D as they have here the B in them, isn't it? With the elimination technique we can easily answer this question. Identify the steps in tricarboxylic acid cycle which does not involve oxidation of substrate. Now tell me, what is tricarboxylic acid cycle? What tricarboxylic acid cycle? What do you mean by tricarboxylic acid cycle? Tell me, children.
Citric cycle.
What? Krebs cycle, TCA cycle, citric acid cycle. What did you say? I didn't hear. Yes, Sangeeta. Tricarbo. So here Krebs cycle is also called Krebs cycle. Krebs cycle is also called tricarboxylic acid cycle, that is TCA cycle. Otherwise it is also called citric acid cycle. So why it is given different names? Krebs cycle because it was discovered and given by Krebs, okay, and TCA, tricarboxylic acid cycle, tricarboxylic acid, otherwise it is called citric acid cycle. Why? Because the first molecule here, when the acetyl CoA with oxaloacetic acid, this is 4 carbon compound molecule, this is 2 carbon. So it will become, what it will become? Citrate. Citrate is the first stable compound formed in the Krebs cycle, citrate or citric acid, So that's why we call it as citric acid cycle. Why tricarboxylic acid? Because citrate, if you see, the formula of it is CH2, COOH, OH and CH2, COOH. Understood? If you see here, how many carbon are here? How many carbon? 1, 2, 3, 4, 5, 6. So it is 6 carbon compound, and if you see COOH here, this is acid group, right? COOH. So we have three of such groups. COOH are 3. That's why we call it as tricarboxylic acid. This is carboxyl group. So tricarboxylic acid, that is citrate. That's why we call it as tricarboxylic acid. So the Krebs cycle is also called as tricarboxylic acid cycle, or it is citric acid cycle because first formed stable product is citrate. Understood? Okay, now coming to the question. What is the question? Which does not involve oxidation of substrate. Oxidation of substrate, it should not involve. Okay. So succinyl CoA to succinic acid. Succinyl CoA. What is the enzyme used here? Succinyl CoA to succinic acid. It is succinyl CoA synthase, in which one molecule of GDP will be converted to one molecule of GTP. If one molecule of succinyl CoA, okay, one molecule of succinyl CoA is converted to succinic acid. Is there any oxidation happening here? No, right? So that would be right. A is the right answer. We will see other options also. Isocitrate to alpha ketoglutarate, it is citrate dehydrogenase is the enzyme used, and in this reaction one molecule of NAD plus is converted to one molecule of NADH. So, anyway, see, addition of hydrogen has taken place. So, this is a reducing equivalent, right? Equivalent. The oxidation of substrate has happened. From the system, the hydrogen is taken by the Na. So, here anyway the oxidation has happened. It is, isn't it? And malic acid to oxaloacetate. So, here the enzyme used is malate dehydrogenase and here also one molecule of NAD plus is converted to one molecule of NADH. Here also the oxidation has happened. Succinic acid to malic acid. Succinic acid will be first converted into fumarate or fumaric acid, fumarate. So, here also it is one molecule of NAD plus is converted to one molecule of NADH. Here also the oxidation has happened. Where the oxidation has not happened is option A, where the oxidation is not happened. Understood? With the first option we get the answer, that is A. And here anyway here GTP is converted to GDP is converted to GTP. Here this is the substrate level phosphorylation has happened. Wait. Substrate level phosphorylation. There are three types of phosphorylation. One is photophosphorylation, which we learn in photosynthesis. In the cyclic phosphorylation, non-cyclic phosphorylation, where the ATP is synthesized. When down the grade, in the redox potential, when down the grade, the electrons were travelling, it synthesized ATPs. In non-cyclic phosphorylation, that is Z scheme, and cyclic phosphorylation, that is photophosphorylation, where light is used. Photo means light is used. Another type of phosphorylation is, that is production of ATP, phosphorylation. Okay. That is substrate level phosphorylation. GTP, it can be converted to ATP. Okay, this is another one, substrate level phosphorylation. Substrate level phosphorylation. Okay, otherwise called as SLP, substrate level phosphorylation. And others phosphorylation is another type. Third type is oxidative phosphorylation. What do you mean by oxidative phosphorylation? At the end, all these reducing equivalents are there. NAD, H, FAD, H2. When these enter ETC, electron transport chain or electron transport system, whatever you call it as, at the end, with the help of ATP synthase, the ATP will be produced, right, with the help of ADP will be converted into ATP. This ATP production we call it as oxidative phosphorylation. Oxidative phosphorylation. Three types of phosphorylation. Understood? Where and all the substrate level phosphorylation is taking place, where the oxidative phosphorylation taking place, and where photophosphorylation is taking place. Okay, if you are understanding, just give me the quick thumbs up. Thank you, Kiran. Yes, we are moving to the next question. We need to choose the correct answer, that is match the following. Citric acid cycle. Citric acid cycle means, okay, where it is taking place? Citric acid cycle is taking place in the mitochondrial matrix. Mitochondrial matrix. Mitochondrial matrix. Glycolysis is taking place in cytoplasm. Electron transport is taking place in the inner mitochondrial membrane. Yes. Proton gradient is formed in the intermembrane space of mitochondria. Any doubts regarding this? That is A2B1. Okay, the first match we got the answer, that is D4 Delhi. So here, if this is a mitochondria, and these are the cristae, the inner mitochondrial membrane has folded itself for maximizing the surface area for these like electron transport chain and the complex to accommodate. So this is the inner mitochondrial membrane. So in inner mitochondrial membrane, what happens? Electron transport chain happens, or ETC or ETS happens. Okay, when this is happening, the H plus, which is in the matrix, this is matrix, will be pumped out to inner mitochondrial membrane. This is inner mitochondrial space, intermitochondrial space, here in between this space, in this space, okay, where the proton gradient will be formed. Understood? And Krebs cycle will be taking place here. The Krebs cycle will be taking place here in the matrix. And if this is a cytoplasm, here glycolysis will be taking place. Glycolysis in the cytoplasm. Understood? Match list 1. Again match the following. Oxidative decarboxylation. Where will oxidative decarboxylation of pyruvate will be taking place? Okay. Oxidative phosphorylation will be taking place in the electron transport system. Glycolysis is also called EMP pathway. Emden, Merhoff and Paranas pathway. 3 scientists who gave this. And oxidative decarboxylation of pyruvate will be taking place. And tricarboxylic acid cycle, the first one is citrate synthase, isn't it? Yes. A2, B4, C3, A2, B4, C3 and D1. So, is the correct answer. Any doubts? Any doubts? How many times the decarboxylation occurs in each TCA cycle? In each TCA cycle, how many times the decarboxylation happens? So, when this is citrate is converted into isocitrate with the help of which enzyme? Aconitase. Isocitrate is converted to alpha ketoglutarate Here the enzyme used is citrate dehydrogenase where a molecule of CO2 will be evolved here and NAD plus will be converted to NADH. Understood? So, here one molecule of CO2 is evolved. One molecule of CO2 is evolved. And alpha ketoglutarate, see here, this is 6 carbon compound, this is also 6 carbon. Now, here as the one molecule of CO2 has evolved, alpha ketoglutarate is 5 carbon molecule, 5 carbon, and this will be converted to succinyl CoA. Succinyl CoA is a 4 carbon compound, which means one carbon molecule again, that is decarboxylation has taken place, and again this is NAD plus is converted into NADH. Okay, again the oxidation has taken place and decarboxylation has also taken place. Here it is alpha ketoglutarate dehydrogenase is a enzyme used. So how many times the decarboxylation occur in each TCA cycle? That is 1 and 2, twice. D4L is a correct answer. 5 carbon compound formed during the TCA cycle is, tell me the answer children, which is a 5 carbon compound? Just now we discussed. Tell me. Should we discuss TCA cycle, the whole cycle once? Shall we discuss that before going for the questions? Yes or no? Just give me thumbs up. So should we discuss then? Ishamita? Social. Okay, B is the correct, alpha ketoglutarate is the correct answer. Is there any lag? Because I feel there is a lag. Subhash, is there any lag? Just check once.
Is there?
Is there any lag?
No. It's clear only.
Yes. Yes, sure. So shall we discuss a TCA cycle once? Okay, we'll discuss a TCA cycle once. Here, from the link reaction, that is, this is pyruvate, is a 3 carbon compound. Okay, it will be converted to acetyl coenzyme A. In here, the enzyme used is pyruvate dehydrogenase. In this step, one molecule of carbon dioxide will be evolved. Okay. Now I am taking only one pyruvate into cycle. Okay, only one pyruvate into cycle. But if you think the glycolysis, one molecule of glucose was yielding 2 molecule of pyruvate. In that I am taking only one molecule of pyruvate here. Understanding? At the end, when we are discussing the balance sheet, I will multiply this into 2, the whole cycle. Now for the easy calculation, let me take only one molecule into the cycle. Okay. Only one molecule into the cycle. Understanding? So here one molecule of carbon dioxide will be evolved here. In this step, oxidation happens, that is NAD plus will be converted into NADH. Understood? NADH. Then one molecule else, coenzyme A is also entered the cycle. Now when this is a 3 carbon compound, one carbon, that is as a carbon dioxide has evolved, then acetyl CoA is a how many compound molecule? How many carbon compound? I am really sorry. How many carbon compound it is? It is 2 carbon compound. Understood? This 2 carbon compound, that is acetyl CoA, now enter the Krebs cycle. How? Enter. So it will be with the oxaloacetate, it will join with the oxaloacetate. This is a 4 carbon compound, 4 carbon. Okay, 4 plus 2, now this will become 6 carbon. 4 carbon oxyl acetate is joining with the acetyl CoA, that is 2 carbon. Now this will, the whole total, it will become 6 carbon molecule, that is citrate. 6 carbon molecule, that is citrate. It is a 6 carbon. Understood? What is the enzyme used here? The enzyme used here is citrate synthase. Citrate synthase. Citrate synthase. Okay, citrate synthase. Now citrate will be converted to isocitrate. It's an isomer of it, nothing much. It's an isomer of it. With the help of the enzyme aconitase. Aconitase. Okay. Now isocitrate will, this is how many compound? No evolution of carbon dioxide has taken place, which means how many carbon molecule it is? It is also 6 carbon. Okay, 6 carbon. Now the isocitrate will be converted to alpha ketoglutarate. Alpha ketoglutarate. The enzyme used here is citrate dehydrogenase. Citrate dehydrogenase. Here decarboxylation reaction takes place, decarboxylation, which means one molecule of CO2 will be coming out of the cycle, evolved, decarboxylation takes place, and one molecule of NAD plus will be converted to one molecule of NADH plus H plus. Okay, NADH, you can call it as NADH. Understood? Then the alpha ketoglutarate will be converted to succinyl CoA. Succinyl CoA, that is succinyl coenzyme A. Here the enzyme used is alpha ketoglutarate dehydrogenase. Dehydrogenase. Even in this, there will be evolution of one molecule of CO2. One molecule of CO2 is evolved. And here, before that, one important thing, the alpha ketoglutarate is a 5 carbon molecule, isn't it? 5 carbon compound. Because it was 6 carbon, 1 carbon evolved, and remaining alpha ketoglutarate is a 5 carbon molecule. From 5 carbon molecule, again 1 carbon dioxide, that is as a carbon dioxide, it has evolved out of the cycle, which means the succinyl CoA is how many carbon molecule? Now it is the 4 carbon molecule. Agree with me? It is a 4 carbon molecule. Agree? Yes. And here in this reaction again, one molecule of NAD plus is converted to NADH plus H plus. Understood? NADH plus H plus. If CoA is here, then there is an addition of CoA into the cycle. CoA is gotten into the cycle. Not much of a significance for now. Okay. Now again, the succinyl CoA is transformed into succinate. Succinate. Here, the enzyme used is succinyl CoA synthase. Succinyl CoA synthase. In this particular reaction, what happens? Substrate level phosphorylation. I said, right, substrate level phosphorylation. That is one molecule of GDP. Okay, GDP. That is guanine diphosphate. Adenosine diphosphate. ATP means adenosine triphosphate. ADP means adenosine diphosphate. Adenosine is adenosine and guanosine. that is same. Okay, it can be converted immediately. This guanosine can be immediately converted. So it is 8, it will be converted to GTP, it will be converted to GTP. So GTP will be converted to GTP, not much of a difference between ATP, GTP and all, fine. Here can we call it as substrate level phosphorylation, because here and all reducing equivalents that has to go into the electron transport chain for them to synthesize ATP at the end as oxidative phosphorylation. Here directly we are getting ATP, that is in the form of GTP. This is substrate level phosphorylation, very important. Okay, please remember, very important. The succinate will be converted to fumarate. Okay, succinate will be converted to fumarate. Here it is, the enzyme used is succinate dehydrogenase, and here also one molecule of FAD plus, it is also a reducing equivalent, will be converted to FADH2. Understood? Succinate will be converted to fumarate with the help of succinate dehydrogenase enzyme, where one molecule of FAD plus is converted to one molecule of FADH2. This is also oxidation. Oxidation has taken place here also. The fumarate is converted to malate. Okay, here with the help of fumarase enzyme. And malate is converted to oxaloacetate with the help of malate dehydrogenase enzyme. Even here also one molecule of NAD plus is converted to one molecule of NADH. Understood? NADH. So this is a Krebs cycle. And oxaloacetate again combined with acetyl CoA to form citrate. Now just remember, I will just tell you where and all the oxidation step is. One of the first oxidation step will be in the glycolysis, where at exactly which step, where G3P, glyceraldehyde 3 phosphate is converted to, tell me the glycolysis, 3-PGA, 3-phosphoglyceraldehyde. Okay, at that first step NAD plus is converted to NADH, that is the first oxidative step. Here you can see next oxidative, this is the second oxidation. Okay, oxidation second, oxidation second. Here if you see again oxidation, third oxidation. Here it is fourth oxidation. Here it is fifth oxidation. Here it is sixth oxidation. Total 6 oxidation steps are here. Understood? How many in one cycle? How many molecules of NAD is produced? In here, 3 molecules of NAD. How many molecules of NADH? Is 3. FADH2 is 1. GTP is 1. If one cycle of Krebs cycle happens. Now, ध्यान से सुनो, pyruvate, here I have taken this whole cycle, one molecule of pyruvate is entering the Krebs cycle. This is Krebs cycle. One molecule of pyruvate is entering the Krebs cycle. Now, each glucose molecule yield 2 molecule of pyruvate. If I into everything, the whole Krebs cycle into 2, if I do, if 2 molecule of pyruvate is entering, then 2 acetyl CoA. If 2 acetyl CoA, 2 oxaloacetate, 2 citrate, 2 isocitrate, 2 alpha ketoglutarate, 2 2 succinyl-CoA, 2 succinate, 2 fumarate, 2 malate, 2 oxaloacetate. If this is there, then everywhere 2 NAD plus, 2 NAD plus, 2 GTP, 2 FAD plus, 2 NAD plus. Everywhere it is into 2, into 2, into 2. If I into 2, then I will do the calculation here. Okay. Now, how many NAD? H? It is totally 6, isn't it? Each NAD yield 3 ATP at the end of the ETC, then it is 6 into 3. The total number of ATP here is 18. FADH2, how many? Into 2, that is 2 into each molecule of FAD yield 2 molecule of ATP. 2 is equal to 4, and each GTP anyway it yield same into 2, that is 2, it is equal to 2. At the end of the ETC, how many molecules of ATP will be getting? 18 plus 2, 20, 24. Total number of ATP this Krebs cycle will be yielding is 24. Understood? 24. This is the balance sheet of the, which one? This is the balance sheet of the Krebs cycle. Now, till here we call it as link reaction. Pyruvate to acetyl-CoA we call it as link reaction, because acetyl-CoA is a link molecule. In amphibolic pathway you can see that from fats also you can yield acetyl-CoA, from protein also you can yield acetyl-CoA. Acetyl-CoA is a link molecule. Understood? In this whole pathway, that is acetyl-CoA is a link molecule. This reaction we call it as link reaction. Even in link reaction we can see 1 NAD plus is, this is in Krebs cycle. This is in Krebs cycle. If you see even in link reaction, how many molecules of NAD is used, NADH is produced? It is 2 molecules, right? So 2 into 3, we got 6 in link reaction. If we see glycolysis, how many molecules? 2 molecules of NAD we yield, which gives 6, and 2 direct ATPs we got, that is 2. Total is 8. So total number of ATP of all these things, if total number of ATP of all these things is 8 in glycolysis plus 6 in Krebs link reaction and 24 in the Krebs cycle. What is the total number? 24 plus 6, 30, 30 plus 8, 38. 38 number of or molecule of ATP we will be getting at the end of ETC. That is, also in the aerobic respiration we will be yielding 38 molecules of ATP at the end. Is it understanding? I hope it is, you understood the Krebs cycle and the balance sheet. If you want I can do the balance sheet in the new page. Okay, we will do the balance sheet. Wait. In the glycolysis, okay, glycolysis, you need to find NADH, FADH2, ATP, total ATP, okay. Total ATP at the end of Krebs cycle, sorry, at the end of ETC. First is glycolysis. How many molecules of NADH we will be attaining? We will be getting 2 molecules, which means 2 into 3 is equal to 6. What is 2 into 3? 2 molecules of NAD. It will be going into ETC, and 1 molecule of NAD will be giving 3 molecules of ATP. 1 molecule of NADH will be giving 3 molecules of ATP, that is 2 into 3. At last we will be getting 6 molecule of ATP at the end of the ETC. FAD, we did not get any FAD, H2. Any ATP, we will be getting net ATP 2. 6 plus 2 is, total number of ATP is 8. In the link reaction, how many molecule of NADH we will be getting? We will be getting 2 molecules. So which means 2 into 3 is equal to 6 we will be getting, and no FADH2 and ATP. So, we will be getting total 6. In Krebs cycle, we will be getting 6 molecule of NADH, which means 6 into 3 is equal to 18. Okay. And how many FADH2? We will be getting 2 FADH2, that is 2 into 2 is equal to 4. How many molecules of ATP or GTP? 2 molecules of GTP, that is 2 into 1 is equal to 2. Total 18 plus 4 plus 2, that is 24. At the end, if you see, the total is equal to 24, 38 molecules of ATP at the end of the ETC, at the end of aerobic respiration. Understood? This is the balance sheet. This is the balance sheet. I hope everybody understood. If you understood, give me thumbs up. Thank you everybody. So, shall we move on to the next question? We will be solving. So, yes. So, we were solving a previous one question, right? The 5 carbon compound formed during TCA cycle is, now tell me, which is a 5 carbon compound which is formed in the TCA? Yes, that is B for Bombay. Alpha ketoglutarate is a 5 carbon compound. Fumaric acid is a, fumarate is a 4 carbon. Oxalosuccinic acid, that is oxalosuccinic acid will be forming in the starting. Okay, we do not want right now this one. Succinic acid is a 4 carbon. Okay, oxalosuccinic acid is a unstable product we will be starting, I mean which will forming at the first. We will not consider it right now, okay, in the Krebs cycle. We will keep it simple. Understood? So, that is a alpha ketoglutaric acid, that is a 5 carbon compound which we can see. The only 5 carbon compound which we see in the Krebs cycle. Rest everything is 6 carbon, citrate is 6 carbon, isocitrate is 6 carbon, then succinate, fumarate, malate, everything will be 4 carbon. Understood? Yes. The number of times carboxylation of isocitrate occurs during the single TCA cycle is, in single TCA cycle, isocitrate, decarboxylation of isocitrate. Isocitrate say we were forming alpha keto glutarate. In this, one molecule of NAD plus is in one cycle, they are single cycle, okay, NADH. In this, one carbon dioxide was evolved here, right. So, how many times the decarboxylation happens? In one cycle, it is 1 time. Yes, 1 time. Very good, isn't it? And here again another one decarboxylation happens, the of alpha ketoglutarate to become succinyl CoA. Understood? We got it. Which of the following statement is incorrect? In ETC, one molecule of NADH plus H plus gives rise to 2 ADP molecules and one NADH2 give rise to 3 ADP molecules. Is it correct? Need to find the incorrect one. One molecule of FADH plus H plus will give 3 molecule of ATP and FADH2 will give 2 molecule of ATP. The first itself is wrong. So, we got the correct answer, that is A. The statement A is wrong, I mean this statement is wrong and we got the correct answer. ATP synthesized through complex 5, yes we know this is correct, this we will go through all the options. And one important thing is, even if you get a correct answer in the first A option itself, just for your confirmation just go through this all other 3 also, okay, because sometimes more than one answer will be correct and we need to find the more correct one. So as we have already went through so many PYQs, you got to know that, right? So for just for the confirmation, we'll go through all the options. ATP synthesized to complex 5, yes. Oxidation reduction reaction produce proton gradient in respiration, yes. During aerobic respiration, the role of oxygen is limited to the terminal stage, yes. Oxygen is a terminal electron acceptor. ऑक्सीजन रोल सिर्फ टर्मिनल में ही आता है. आफ्टर कॉम्प्लेक्स 4 द इलेक्ट्रॉन्स फ्रॉम द cytochrome A3 molecule, from cytochrome A3 molecule, the electrons will be given to oxygen, okay, with the help of 2H plus it will become water. So role of oxygen terminal hai, so this is also we got it. So D is also right. So the wrong option was A. The number of substrate level phosphorylation in one term of citric acid cycle. The number of substrate level phosphorylation. Tell me. Substrate level phosphorylation GTP बन रहा था. कहाँ बन रहा था? Succinyl CoA when it converted into succinate. In the presence of succinyl CoA synthase, यहाँ बन रहा था substrate level phosphorylation. This is direct ATP or GTP was synthesized. That is called substrate level phosphorylation. Very good. One time in one turn. Everywhere whenever you are reading especially the photosynthesis in plants, photosynthesis in higher plants and respiration in plants, hamesha this one, how many turns they have given? It is 1 turn, 2 turns, 3 turns, how many turns they have given? Mark kar do. Understood? If they would have given 2 turns, then it will be how many molecule? Number of substrate level phosphorylation, that will be the number of substrate level phosphorylation. The number of substrate level phosphorylation will be 1 only, but molecules will be producing, GTP will be producing, we will be producing 2 molecule of GTP. So they can ask question in any form, any format. So just for a better this thing, we'll mark it, how many turns they have given. Pyruvate decarboxylase, pyruvate dehydrogenase activity during aerobic respiration requires. When I was telling you about pyruvate converting into acetyl CoA, when the pyruvate is converting into acetyl CoA, the enzyme here used is pyruvate dehydrogenase. Actually this is a complex, okay, it's a complex, many things come into picture. One of that is the cofactor here used is magnesium. Understood? One of the main thing here used is magnesium ion. Magnesium as a cofactor it is used, okay. Again entry of acetyl CoA, removal of carbon dioxide, everything takes place. NAD plus will be converted to NAD H plus H plus, okay. So this is a thing. So what we require? We require magnesium as a cofactor. Pyruvate dehydrogenase require magnesium as a cofactor. What is the role of NAD plus in cellular respiration? Tell me. What is the role of NAD plus? Okay, it functions as an enzyme. No, it is not an enzyme. It functions as electron carrier. Yes, it definitely. And it is a nucleotide source for ATP synthesis. Nucleotide source. No. It is a final electron acceptor for anaerobic respiration. No. So why it is it's a function as electron carrier? How? Actually NAD plus, which will be converted to NAD H plus plus H plus, H plus H plus. They receive electron Understood? They take up electrons from the system as well as they take up H plus ions from the system. So when they take it, okay, they remove the H plus from the system, which means oxidation happens in the system. They themselves undergo reduction. Okay, receiving electron, they receive electron and they undergo reduction. Okay, so they themselves undergo reduction. In the process they take up electrons from the system, and these electrons ultimately they what they will be doing ultimately in the electron transport chain, the first thing, that is the first complex 1, otherwise we call it as NADH dehydrogenase. This complex we call it as NADH dehydrogenase. This NADH, which now has electron as well as protons, this undergo again here, they give the electrons into 2 electrons into the complex 1 and they convert into NAD plus here. Whatever these were the reducing equivalents, we call it as reducing equivalents. They undergo, it received the electron and they themselves underwent reduction. Now they undergo oxidation, they give away, the removal of electrons takes place here. These electrons will be given to the complex 1. These electrons will be taken by FMN at first, flavin mononucleotide. Okay, FMN, and from FMN it will be given to iron sulfur complex. Then it will be given to coenzyme Q, that we call it as ubiquinone. Understood? Ubiquinone. These 2 electrons will be given to here. So this is the starting of the electron transport chain. Understood? So they function as electron carrier from the system, from wherever, it may be in the matrix, maybe it in the glycolysis where it was happening, in the cytoplasm. They took the electrons and ultimately they are given in the electron transport chain. This electron transport chain, it is happening in the inner mitochondrial membrane. Understood? So now ultimately they had to give this electron to electron transport chain. So what their function was? Their function was whole total electron carriers. Understood? Which of these statement is incorrect? We need to find the incorrect option. Enzymes of TCA cycle are present in mitochondrial matrix? Definitely, because TCA cycle is happening in mitochondrial matrix. The enzyme should also be in the mitochondrial matrix. Glycolysis occurs in cytosol, that is cytoplasm. Yes, we know that, basic. Glycolysis operates as long as it is supplied with NAD that can pick up hydrogen atoms. Yes, if NAD is not there, reducing equivalents, there is no proceeding of the process. Oxidative phosphorylation takes place in outer mitochondrial membrane. Oxidative phosphorylation actually takes place in the inner mitochondrial membrane. Inner mitochondrial membrane. All complexes, complex 1, 2, 3, 4 and the 5th one, that is ATP synthase, all are present in inner mitochondrial membrane only. So the option D is wrong. Are you getting it? Which statement is wrong for Krebs cycle? Tell me which statement is wrong. There are 3 points in the cycle where NAD plus is reduced to NADH plus H plus. 3 points? Yes. There is 1 point in the cycle where FAD plus is reduced to FADH2? Yes. During conversion of succinyl CoA to succinic acid or succinate, a molecule of GTP is synthesized? Yes. The cycle starts with condensation of acetyl group, acetyl CoA, with pyruvic acid to yield citrate. Was it pyruvic acid, the acetyl that is acetyl group, that is acetyl CoA, was combining with oxaloacetate, was combining with the oxaloacetate, which is a 4 carbon molecule. Acetyl CoA is a 2 carbon molecule. So 4 plus 2 became citric acid, that is 6 carbon molecule. है ना? Pyruvic तो 3 carbon molecule है, and pyruvic acid was converted into acetyl CoA. So this is wrong, right? So D is wrong. We need to find the wrong statement, that is D. D for Delhi is a wrong statement. Why it is wrong? Because acetyl CoA, which is 2 carbon molecule, is combining with oxaloacetate, that which is 4 carbon molecule, to yield citric acid, that is 6 carbon molecule. 4 plus 2 is 6, not with the pyruvic acid. Oxidative phosphorylation is addition of phosphate group to ATP, addition of phosphate group to ATP, no. The formation of ATP by energy released from electrons removed during the substrate oxidation, yes. The formation of ATP by transfer of phosphate group from the substrate to ADP, formation of ATP by transfer of phosphate group from the substrate to ATP, no. ATP, substrate to ATP, no. We need to find the oxidative phosphorylation, not this one. Oxidation of phosphate group in ATP, no. It is a formation of ATP by energy released from electrons removed during the substrate oxidation. What do you mean by this? Actually, formation of ATP by energy released from the electrons removed during the substrate. So wherever the glycolysis or Krebs cycle, wherever it was taking place from the substrate, the electrons were removed and given to what? Reducing equivalents of NADH plus H plus, NADH plus H plus, or FADH2. The electrons were given to these from the substrate, the electrons were given to the reducing equivalents, these two. Okay. Then these underwent electron transport chain, where they gave up, they given the electrons to the ETC, that is electron transport chain, where ATP was synthesized at last with the help of ATP synthase enzyme. So B is the correct answer. Formation of ATP by the energy released from the electrons removed during the substrate oxidation, right? Cytochrome are found in, where the cytochromes are found? Cristae of the mitochondria, lysosomes, matrix of mitochondria, or outer wall of mitochondria. Cytochromes are found in the cristae, that is cristae is the inner mitochondrial membrane. Inner mitochondrial membrane. This is where the cytochromes are found, isn't it? In the ETC. So A is the correct answer. Fatty acids are connected with the respiratory pathway through, I told you, right, there is a linking molecule, that is acetyl CoA. From the fatty acid, beta oxidation takes place. Beta oxidation of fatty acids would give rise to the acetyl CoA. Understood? That is a linking molecule. From everywhere, even from the this thing, protein, we will be getting acetyl CoA. Acetyl CoA is a linking molecule. Match the following. Porins. Porins means pores. You can see pores. Huge pores in the outer membrane of mitochondria. Huge pores in the outer membrane of mitochondria. Leg hemoglobin, it has a pink colored nodules. H plus accumulation takes place between in the lumen, yes. This is a photosynthesis chapter, lumen of the thylakoid. And respiration is a amphibolic pathway. A4, there is only A4 in one option, that is C. We got the answer with the first matching. Which of the following biomolecules is common to respiration mediated breakdown of fats? carbohydrates and proteins. Now tell me which is the common, that is a linking molecule I was always from the starting of the lesson we have been discussing about, that is acetyl-CoA. Which of the following biomolecule is common in the respiration mediated breakdown of fats, carbohydrates and proteins? Acetyl-CoA, easy. Respiratory quotient value of tripalmitin is direct NCERT question, that is 0.4, sorry 0.7. 0.7 is the correct answer. So, this is the end of the PYQs. Any doubts till now? Any doubts, children? If there is any doubt, please you can on your mic and ask me, otherwise put it in the chat box. If you understood everything, you can give a thumbs up. Thank you, Kiran. Thank you, Shamita. So, okay, if there is no doubt, we will end the session here. So, thank you so much everybody. So, the class ends here. You can leave the call.